Hello everyone, welcome back to Alchemist. Our topic is the ubiquitous regulatory role of calcium ion in the muscles concentration. The word ubiquitous is key here because calcium ions are present everywhere in biological systems, acting as a vital messenger that tells our muscles exactly when to pull and when to let go. Before we dive into the details, let's look at the key stages of our journey today. We will analyze this biological process through five specific chemical lenses. First, we will examine the coordination chemistry of calcium. We will discuss why its unique ionic radius and charge density make it the perfect candidate for rapid signaling compared to other metal ions. Next, we move the protein level with the EF hand motif. This is the molecular sensor, a specific structural shape in proteins that acts like a specialized glow to catch and hold calcium ions. Third, we will look at the regulatory mechanism. This is the master switch, where the binding of calcium actually triggers a structural change, clearing the path for muscles fibers to interact and contract. In the fourth section, we will ask the fundamental question: why calcium? We will compare calcium ions to other ions like magnesium or potassium to understand the evolutionary and chemical advantage of using calcium for this specific job. Finally, we will discuss homeostasis and sequestration because too much calcium can be toxic to a cell. We will explore how the body stores it away and pumps it back out to allow the muscles to relax and reset for the next moment. With this roadmap in mind, let's start with our first topic, the unique coordination chemistry of the calcium ions. On the top left, we see calcium preferred environment. Unlike many other metals, often adopts an eightfold coordination geometry, it has a strong preference for oxygen donors, specifically from amino acid residues like aspartate and glutamate, as shown in the HSAB theory section. Calcium is a hard acid, meaning it seeks out hard base like oxygen to form stable but flexible bonds. A major challenge in the cell is selectivity. There is much more magnesium in the cell than calcium. However, as the energy graph shows, calcium has a larger ionic radius. Proteins are geometrical designed to provide a perfect pocket for this larger radius. Magnesium is simply too small to coordinate effectively in these specific sites, allowing calcium to bind even when its concentration is low. Look at the bottom left. Calcium is structurally flexible, with coordination numbers ranging from 6 to 8. This flexibility leads to a high ligand exchange rate. It can bind and release on and off extremely quickly. In the EF hand motif, this causes a rapid protein refold or snap, which is the physical movement that moves the troponin complex and starts the concentration. Finally, we see the allosteric switch mechanism because calcium can move across membrane through specialized ion channels so quickly. It creates a near instantaneous concentration gradient. This triggers a conformational change in the muscle filaments, acting as the final go signal for the concentration process shown in the bottom right. Next, we will focus specifically on that EF hand sensor. On the top left, we see the EF hand structure. It consists of two helix, the E helix and the F helix, connected by a flexible loop. In its inactive state, the loop is open. However, when calcium enters, it triggers the active state. Notice the ligand like aspartate and glutamate, they provide a precise geometry that coordinates the calcium ion, causing the helix to reorient. A critical feature shown in the top right is how the motif chooses calcium over magnesium. The loop is pre-organized for an optical sevenfold coordination. As the energy graph illustrates, magnesium ion is too small for this specific geometric constant. Even though magnesium is more abundant in the cell, the EF hands pocket is tailor-made for the larger radius of calcium ions. Now, look at the bottom left to see the allosteric switching mechanism. When calcium binds, it causes a conformational change, often described as a T to R transition. This shift is a hydrophobic patch on the protein surface. This is not just a shape change, it is a signal. This exposed patch acts like a molecular velcro that binds to other proteins such as troponin T and I. Finally, the bottom right illustrates the result. Kinetic control because the calcium ion channel permeability is so high. The response is near instantaneous. The allosteric change leads to interdomain interaction that allow for cross bridge formation. This is the exact moment the muscle's fiber receives the command to contract. Now, let's see how this switch works within the larger regulatory mechanism. On the left, we see the inactive state. In this resting condition, the protein tropomyosin acts like a physical barrier. As you can see in the zoomed in box, it hides the acting binding sites because these sites are locked. The myosin hits cannot attach and the muscle remains relaxed. Notice troponin C in its ribbon form. It is waiting for the signal, effectively inhibiting the path of myosin. Now look at the right side, the active state. When calcium binds to the EF end of troponin C, a snap occurs. The energy graph for allosteric efficiency shows that the calcium ion bounded state is energetically favorable, triggering a rapid T2R like transition. This is not just a chemical change, it is a high speed mechanical shift. This binding triggers a specific conformational switch. Look at the interaction between the aspartate and glutamate residues. This molecular refolding exposes the protein surface, causing the troponin tropomyosin complex to shift its position. This shift finally unlocks the acting myosin binding sites that were previously hidden. The final result is shown at the bottom. Kinetic output with the binding sites exposed. Acting myosin interaction can finally take place. The myosin heats bind to acting, forming cross bridge that slide filament past each other. This is the exact moment the chemical energy of a concentration gradient becomes the physical force of a muscle concentration. Next, we will discuss specifically why evolution chose calcium for this vital role over other ions. First, let's look at the hard ligand preference. As we can see in section A, calcium is a hard acid that thrives on hard interaction with oxygen donors like aspartate and glutamate. The affinity graph below clearly shows that while calcium has a very low affinity for soft ligand like sulfur or nitrogen, it has a massive rapid affinity for hard O donors. This allows it to bind specifically to oxygen rich proteins loops even in a crowded cellular environment. Second is conformational plasticity. Calcium is unique because it can exist in multiple coordination spheres, allowing for both compact high affinity states and flexible multi ligand states. This flexibility allows the troponin C protein to undergo a specific conformational switch. The energy graph shows that the refolding energy for calcium bond states is much more efficient than magnesium, allowing for a dramatic T2R like transition that exposes the protein surface for new interaction. Finally, we have first kinetic and the concentration gradient. In section C, we see that calcium is stored at high concentrations in the sarcoplasmic reticulum. When the signal is given, it rushes down the concentration gradient into the cytosol where the concentration is very low. Crucially, binding speed is much faster than the released speed, creating a rapid allosteric state transition. The final result is shown in the bottom right graph, a rapid muscles activation cycle. The sharp spike in intercellular calcium is followed almost instantly by a spike in physical force. This instantaneous link between a chemical gradient and mechanical output is why calcium is the perfect signaling ion for high speed muscle responses. We will look at how the cell maintains this delicate balance through homeostasis. In the center, we see the release mechanism. When a nerve impulse arrives, ryanodine receptors, the SR calcium channels open. This triggers a massive signal amplification because of the established gradient. Calcium rushes out to bind with troponin C, causing the allosteric state transition we discussed earlier. The balance between this rapid release and the SERCA pump's steady removal is what we call hemostasis. On the top right is a vital protein called calciquestrine. Think of this as the cell's calcium warehouse. It is a high capacity binding protein that uses multiple coordination spheres to hold onto large amount of calcium. This buffering ensures that the concentration of free ions inside the SR doesn't get too high, while still keeping a massive supply ready for the next controlled release through a specific conformational switch. Finally, look at the bottom right graph. You can see the calcium signal amplification cycle. The intracellular calcium level spikes to generate force, but it must drop just as quickly for the muscle to relax. This rapid drop is the result of the SERCA pumps. Calciquestrine working together to clear the cytosol. To conclude our presentation, let's bring all these pieces together. We have moved from coordination chemistry to molecular sensors, and now we see the entire integrated calcium regulation network in action. It all begins with the neural trigger. Initial deep polarization causes a fast release gradient of calcium ions. As these ions reach the troponin C allosteric switch, we see that snap we discussed a conformational T to R transition. This is where the chemistry of the calcium ion directly detected the shape of the protein. This signal then undergoes kinetic amplification. As shown in the center, the calcium signal propagates through ryanodine receptor release channels, leading to a massive increase in local concentration. The energy graph for a single switch reminds us that this process is governed by integrate force calcium kinetics, where the amplitude of calcium spike determines the strength of the concentration. The arrows point us to the ultimate goal, mechanical output. The refolding of the regulatory proteins exposes the surface for actin myosin interaction. This leads to the power stroke and sarcomere shortening, which is the physical concentration of the muscle fiber. This actin myosin cycle is the direct result of the chemical go signal provided by calcium. Finally, we cannot ignore the SERCA pump and their reputate mechanism. For the muscle to reset, we need ATP driven reputate. Proper hemostasis is not just about movement, it is about overall cell health. Incorrect signaling can lead to toxicity of metabolic failure. Thank you for listening. It was a pleasure presenting to you all today.